Lior. I'm Lior from the Weizmann Institute. I'm going to talk about the convergence between cancer, aging, and inflammation. Okay, so the median age for cancer diagnosis is about 65 years of age. So it's very important to understand the age dependency etiology of cancer. So in order to tackle this important question, this big question, we decided to ask what will happen if we activate an oncogene, a driver of tumorigenesis, in the normal age tissue of an old mouse. We choose a mouse model for skin squamous cell carcinoma. It's a non-melanoma skin cancer, which has two risk factors. One is, of course, sun exposure, and the other one is age. It's a mouse model that was developed by Paul Cavari from Stanford. Basically, it's an HRAS fused to the estrogen receptor. It's expressed under the K14 promoter. It's expressed in the basal layer of the skin epithelium. And I'm going to share results of mice treated for 1 month and 3 months. The transgene is activated via topical application of 4-hydroxytamoxifen. And you can see that when we activate the RAS for 1 month, we see massive hyperplasia in both young and old mice. And so we have massive hyperplasia in both young and old mice, but the old mice show much more levels of dysplasia. And I'm not sure if you can see it with this slide, but we see accumulation of senescent cells, as can be detected by the senescence-associated Pelagal marker. And there are more senescent cells accumulated in the aged tissue. And interestingly, those cells accumulate in the dermis and not in the epidermis where the RAS is activated. So one thing that we know about accumulation of senescent cells that they can basically drive tumorigenesis if they are not, as Valerie is told us, they can drive tumorigenesis if they are not evicted by the immune system. And indeed, if we activate the RAS for 3 months, what we see is in about 50% of the aged mice, they develop in situ squamous cell carcinoma. It's not yet an invasive type of carcinoma, but it is, afterward, a carcinoma. And we see much more, even much more senescent cell accumulated in the dermis of the old tissue compared to the young tissue. Where in the young tissue we see strong activation of hair follicles and hair growth. If we look at global gene expression of the whole skin after 3 months of RAS activation, we see in the aged mice a strong expression of many chemokines and many genes that are associated with mast cells. And if we stain with toluidine blue, which is staining for mast cells, we see strong accumulation of mast cells, and we believe that it is somehow, we don't know yet how, but it is associated with the accumulation of the senescent cells. And one last thing, when we activate the RAS for 1 month, and then we can stop activating the RAS if we withdraw the tamoxifen. What we see that the young tissue quickly recovers and regenerates. It turns from the strong hyperplasic tissue into almost normal type of skin, but the aged tissue remains very much hyperplasic. And again, we see that when we stain for senescence, most of the senescent cells disappear in the young tissue, but many senescent cells are retained in the aged tissue. And we believe that this is contributing factor to the to the high level of hyperplasia in the H tissue. Some conclusions and mostly questions. So oncogene activation in H skin results in increased inflammation that is associated with senescent cells. And it also is correlated with advanced tumorigenesis. We think that the senescent cell turnover is slow in H tissue and could be associated with what is called immunosenescence, the decline in the immunocompetence of H organisms. We see accumulation of mast cells for much larger extent in the H tissue, and this is again is a possible driver of tumorigenesis. And to conclude, possibly that the oncogenic activity alone is not sufficient to induce tumor formation, as we could see that it does not form tumors in the young mice. It could be that the H microenvironment is a sort of a fertile ground to tumor formation. That's it. Thank you to all the people that are here. Thank you.
Any questions?
Okay, thank you.
Thank you. So the presenter is Alisa Gif from Weizmann, from Lior Hospital. And your topic is regulatory mechanisms and mediating results.
Thank you very much. So as you heard earlier from Valerie, the senescence program plays an important role in maintaining normal physiology of wound healing, at least in liver fibrosis involving the NK-mediated killing of senescent cells. Therefore, in my research, I try to find how NK cells specifically target senescent cells, focusing on the killing mechanisms, and to try to assess the impact of this process on liver fibrosis as a model for the tissue damage response. So by which mechanism NK cells kill senescent cells? In order to answer this question, we employed an in vitro cytotoxicity assay where we plate normal growing and DNA damage-induced senescent cells in the same density, add NK cells, and by performing a viability test, we can determine the percent of cytotoxicity. It goes automatically. Anyway, performing this assay, we determined that NK cells can specifically target and eliminate senescent cells twice as efficiently as growing cells. So we used this system further in our research. So NK cells can actually kill target cells through two independent pathways. The first being the death receptor pathway, where the death ligands FASL and TRAIL can trigger caspase-dependent apoptosis in target cells. The second pathway, the granulocytosis pathway, involves the secretion of perforin and granzyme B containing granules upon interaction with the target cell. Perforin is responsible for perforating the target cell membrane, enabling the release of granzyme into the target cell, where it can induce both caspase-dependent and independent apoptosis. Both pathways have specific inhibitors, therefore we can evaluate the specific contribution of each pathway to the senescent cell killing. So can a senescent cell undergo death receptor killing? To answer this, we added TRAIL. And as you can see, TRAIL induces efficient killing of both growing and senescent cells. However, senescent cells are somewhat more resistant to the TRAIL-induced cytotoxicity. Similarly, if you look at the white bars, the second death ligand, FASL, can induce efficient killing of both growing and senescent cells. And by adding caspase inhibitors, we can see that this FASL-induced cytotoxicity is attenuated. So we know that senescent cells can undergo death receptor-mediated killing. However, the really interesting question is whether this pathway contributes to the NK cell killing of senescent cells. So we added the caspase inhibitors, and afterwards we added NK cells, but to our surprise, this did not attenuate the NK cell specific killing of senescent cells. Therefore, actually, we can conclude that the death receptor pathway is dispensable for the NK cell targeting of senescent cells. In light of this result, we now took a pharmacological approach to evaluate the contribution of the second pathway, the granule exocytosis. So again, we used two specific inhibitors for this pathway, and what we found is that in this case, the killing of senescent cells was dramatically reduced, both within NK cell line and primary NK cells. As we already know, the killing of senescent cells in liver fibrosis limits, in the liver fibrosis model, limits the extent of fibrosis and facilitates tissue repair. Therefore, we were very interested to see if in this physiological relevant cell type, the killing is done through the granule exocytosis pathway, and the answer is yes. Thereby, we can conclude that the granule exocytosis is actually plays a general role in the immune surveillance of senescent cells. So naturally, the first question that comes to mind is what determines this pathway bias towards the granule exocytosis. And we actually looked for the answer in the recently discovered oncogene-induced senescent marker, the decoy death receptor DC2, which is a p53-induced gene and is one of the trail decoy receptors. It suppresses trail-induced apoptosis since its intracellular domain is truncated and effectively acts as a competitive inhibitor to trail-induced apoptosis, and its contribution to the senescence program has been unknown. This immediately motivated us to check the expression level of DC2 in our system in DNA damage-induced senescent cells, and as you can see, both in the RNA level and the protein level on the surface cell membrane is largely elevated. Now, since DC2 is a competitive inhibitor of death receptor ligand, we thought that this could explain the pathway bias. So to check this directly, we transiently and stably knocked down DC2, and we found that this results in a higher NK cell cytotoxicity towards senescent cells. So therefore, we can conclude that NK cell cytotoxicity towards senescent cells is attenuated by the upregulation of DC2 in senescent cells. Now, if the contribution of the granule exocytosis pathway is important in vivo as we saw in vitro, then this should altering or inhibiting this process in vivo should alter the progression of liver fibrosis, since in this pathology, the killing of senescent cells facilitates tissue repair. So we induced liver fibrosis in peripheral mice. And what we found is that indeed there's an elevation in the scarring area, which is accompanied by expansion of the hepatic stellate cells. Now, do these cells become senescent? The answer is yes. We have an elevation of senescent activated stellate cells in the areas adjacent to the scar and within the scar, and an elevation in DC2. Therefore, we can conclude that the excessive liver fibrosis can arise from the failure to eliminate activated hepatic stellate cells once they become senescent when we actually disable the granule exocytosis pathway. So, in conclusion, NK cells specifically recognize and kill senescent cells both in vitro and in vivo. It is the granule exocytosis pathway which mediates the NK-mediated surveillance of senescent cells. The pathway bias can be explained by the upregulation of DC2, both in vitro and in vivo. And in the mouse model of liver fibrosis, peripherin-mediated clearance of senescent activated HSCs is crucial for tissue repair. Thank you. I would like to thank my supervisor, Valerie, and my collaborators.
Questions? No questions? Okay. Thank you. The next presenter is Anat from Ben-Gurion University from
Healing and compromise. I only have 5 minutes to explain something that should take at least 15, so I apologize in advance for the explanations. The research question was very simple: is the longevity phenotype good for wound healing? And what chose the skin as a model for various reasons, which I don't really have time to explain. Wound healing was part of the research question, and I will explain briefly why we chose alpha-mupa mice. You already heard about them a bit before, so I will make it brief. They contain the UPA transgene attached to the alpha crystalline promoter, which should be expressed only in the ocular lens in the eyes, and that does happen, but without any effect, any real effect. What does happen unexpectedly is an ectopic expression in the trigeminal nucleus of the brainstem. The cause and effect is not clear, but there is some behavioral change. You already heard about their different circadian rhythm regulation. They also eat less, and maybe as a result they live longer. This is data from Freud and Wiese. You already saw pictures of how the mice look when they're old, the difference. I did a horrible thing to these mice. I did a full thickness wound at the tip of the skull, which shows the tip of the skull because most wound healing assays are done on the back or on the hind legs, but over there it's contractile. The skin just contracts around the wound, and the skull has a splinting effect which does not enable the wound to contract, so it's a replicative repair or cell migration basically. And we followed up on the wound closure, including taking samples for histology and transcriptomics. And obviously we compared alpha-mupa and wild type, and we also prepared young and old animals. All are 2-year-old animals, over the median lifespan. This is how the wound healing looks. The important thing to take from here is that at day 21, all wounds are fully closed. You see full fair coverage, completely shut. When you look at the wild-type animals, you can see that the old wild-type animals take a lot longer to close the wound than the young ones. And when you look at the alpha-mupa mice, you see there is no difference between the old and the young. When you look at all of it together, you see that it's not that the alpha-mupa mice heal the wounds faster than the wild-type mice, it's just that they retain the speed of wound healing up to an old age. Surprising thing was the histology samples showed that the quality of wound healing was exactly the same in all groups, all the young alpha-mupa and wild-type. The quality of wound repair is the same. There may be some minor differences, but overall, it's the same scar tissue in all the groups. This was a bit surprising. The first conclusion is all roads lead to Rome, meaning it doesn't matter how long it takes the animals to get there, it's still the same quality of wound repair even in the old age. Try to compare the data to other studies on transgenic mice with altered lifespan, and at first glance we didn't see any correlation between the lifespan of the mouse and the speed of wound healing. When we isolated only wound healing studies done on older ages, you see an almost perfect correlation. Again, meaning that you see the effect of the longevity phenotype only on older ages. You don't see it in the young. There's some sort of threshold in which the delay in wound healing starts to take effect. So slowly but steady wins the race, meaning it's not important how fast the wound closes at the young ages. The important thing is to retain the speed of wound healing up to an old age. As I said, we sent some of the samples to transcriptomics. The first interesting thing that we saw, even though the histology samples show that the skin is overall the same, you see a very large number of differentially expressed genes in the wild-type mice on day 0, before the wound is even done. A very large number of differentially expressed genes between the old and the young, and a very low number of differentially expressed genes in the alpha-mupa mice, between the old and the young. In the control skin, meaning even though the histology sample looks overall the same, there is some sort of undergoing processes which are different. And this is the same for day 7 and then day 21 after wounding. See very large number of differentially expressed genes in the wild-type mice between the old and the young, and very low number in the alpha-mupa mice. Especially interesting is that when we looked at day 21 and we compared old and young wild-type mice, you see an upregulation of several growth-promoting pathways. again, I'll remind you that day 21, the wound is fully closed. And still there is an upregulation of growth promoting pathways. And we do not see that in alpha-mupa mice. This is just an example of the mTOR pathway. There are several others. For example, it's the same with the TGF-beta signaling pathway, only overexpressed in the wild-type mice on day 21 and the old one. So basically, we think even though the wound is closed, there's still some process going on of growth-promoting nature. We think maybe the wound healing process did not completely shut down. There's still something going on beneath the surface. We don't see it in histology. That's it. That's our growth.
From Gurion University, Anat by the Swiss lab, and Anat over there.
Sadly, Neta is actually right now in the United States. Some combination of honeymoon and doing science. So, all these pictures and this stuff. So, where I want to start with you is protein misfolding diseases. We are sort of related to them during the meeting, and we talked about the fact that there are many diseases that are associated with aging. Some diseases, the most or the best predictor of their onset is age. And those are the family of protein misfolding diseases and neurodegenerative diseases such as Parkinson, Alzheimer's, etc. What's interesting about those diseases is that they also share, even though they affect different proteins, different tissues, they also share common features, which is accumulation of damaged proteins with age. And so when me and Neta were developing this project, we were thinking, how can we sort of think about it and think about the processes that occurs during aging. And we sort of think that, you know, we call it the organized versus messy room theory, where we basically starts here, it's the organized room, everything is functional, everything is reachable, and everything is well functioning, whereas in the old organism, there is a mess, there are aggregation accumulating either in specific places or other places, the equipment is old, things are not really running as they should. And so then we ask, how do we get from here to here? And almost as an assumption in the field is what we call slow accumulation of damage over age. So we assume that if we see disease in old age, that means that we get there because we sort of don't take care of things, and then eventually we get there. So one day I don't put things in place, and then over time things get messy. If you have toddlers at home, you know that you can get mess very quickly. You don't need a lot of time. Okay, so you can actually turn your back 1 minute, and then when you turn back to that room, it's very, very messy. And one of the things that I think people are not considering between going to slow accumulation of damage or fast accumulation of damage is not just how fast damage accumulates, but also how fast damage is removed. Okay? So this is a steady state reaction where we have something that causes damage over time, protein misfolding, and then we also have a lot of processes in the cell that clear damage, and we heard about those processes today. So those are chaperones that can correct protein damage, those are the proteosomes that can clear damaged proteins, and the autophagy system that can clear damaged proteins, and they work in synergy to keep the cell functioning. So we might in fact have in a young organism a lot of damage, but then also very, very effective protein folding machinery that clear that damage. And that system might not function the same in old animals and young animals, and it might not be completely relevant to the rate of damage, or it might be something in between. Of course, we like to think in extremes, so that helps us sort of figure out what we're doing. So we sort of define the question of whether or not proteostasis, and that means all those systems that take care of the proteins in the cells, whether or not their function decline with aging. One of the systems that we used as a model system, because it's very attractive to study, is the heat shock response. And why is it a model system? Because when we apply stress, we cause misfolded proteins. This causes a transcriptional response where HSF1 goes to the nucleus, produce new chaperones, and when we get attenuation of the response, this response, the ability of the organism to activate this response will influence their survival under heat shock conditions, okay? So we can ask whether or not the activation of the heat shock response is similar in young animals versus old animals, and that will tell us how the ability of the animals to respond to stress changes. And so we decided to go along the tool and start looking at the aging of the animals, and you have to start somewhere, so we started with young animals. And that's day 1 of adulthood. And we did a heat shock response, and we calibrated it so that 70% of the animals under the heat shock condition will survive. It's very simple. You take worms, you heard about them, put them in a plate, put them in a bath, heat them to 37 degrees, come back after 6 hours and ask how many are surviving. And we have a condition where 70% survive. And then we do the same experiment 24 hours later. And to our surprise, already at day 2 of adulthood, and those, mind you, are fertile, vigorous adults, they don't survive the same stress. Those are not aged animals. Those are they're age-matched animals. 24 hours later, don't survive the same stress.
They get it twice, or they
No, it's naive animals. So that's one of the advantages of working with C. elegans is that those are clonal population, and so they grow on the same plate. You take a bunch of them. Of course, you repeat it many, many times. This is what you get. And it's consistent. So then we asked, if we break it down, how close can we get? And in fact, if you look at it, basically, somewhere between 12 hours, 7 hours, and 19 hours, things change. So this is not a process where something slowly accumulates, or we have some kind of damage that accumulates over time. This is almost like a switch. One moment, everything is fine, we have a heat shock response. The next moment, we don't have any heat shock response anymore. Something completely changed in the way those animals process the environment and respond to it. What happens to C. elegans at that time point? So we go through embryogenesis, larva stages. This is our young adult. It just reached adulthood, it still doesn't make embryos. This is the full adult. And notice the difference of 8 hours between being young adult and being a fertile adult, which is exactly the point where we see the difference. So this was screaming at us to check whether or not fertility and basically reproduction has anything to do with the response we're seeing, and if the regulatory mechanism is linked to reproduction. And so you heard already that one of the pathways that can increase longevity or affect longevity in C. elegans is the reproductive pathway. And that particular pathway works that if you kill the germline progenitor cells and you don't have germline, then you extend lifespan. But if you kill the entire reproduction system, you still have wild type longevity. So it's something about the germline and whatever the signals that they are producing that affects lifespan. So we started using the mutations, every mutation in the field. We did the GON, this is without any gonad, without any reproduction system, we get the same thing. We did without sperm, without oocyte, we didn't get anything. But when we look at the germline, we get now rescue of the protein folding, the heat shock response. Mind you, again, this is not when we're talking about differences in aging, because those animals, the difference between them is 24 hours. They're not aged. Those are young adults. But now they actually maintain the ability to activate the heat shock response. We can see that this is the ability to no pictures. The ability to activate heat shock response by using a GFP reporter, transcription reporter, and this is quantification of the GFP reporter over time. And while the wild type basically collapse, you don't have a heat shock response after day 1 of adulthood, the germline animals maintain it over 2 weeks, which is almost the time that they will survive. So at that point, they start dying. So what we're saying is that we have robust proteostasis in the young animals, and then there's some kind of a signaling from the germline, closes this, and then we have limiting proteostasis. If we remove the germline, the switch is on, and we have robust proteostasis. What are the implications? We were talking about many things that are associated with aging, and this is a very early event. So we talked about sarcopenia. It was presented. Sarcopenia can be shown in C. elegans just as well as humans. And basically what you see is you see very nice myofilament. This is myo3 GFP. And you see a very nice myofilament organization of a muscle cell. This is 1 muscle cell, 2, 3. And day 1, they're organized. Day 3, you start seeing something. Day 5, it's increasing. And by day 7, you have misorganization of myofilament. The animal is okay at this point, but basically as soon as as you hit this mark, you start seeing paralysis in those animals. Okay? If we now take the germline-less animals, and we did it with glico, we did it with other mutations, you have very nice myofilament over age, and you also slow down paralysis. So one of the consequences of changing the way the organism deals with protein folding and heat shock response early on in adulthood is changing the rate of aging and sarcopenia. If we look at protein misfolding diseases, and we have models in C. elegans, we have protein aggregation models where you can see an age-dependent aggregation in those models and age-dependent toxicity. If we put it in the germline-less animals, you lose the aggregation and you also reduce toxicity dramatically. So we can look at a beneficial effect much later in life to this very early change in the ways the organism responds to stress. One of the most important things is that we're talking about somatic changes in the way protein folding occurring, but the mutant or the change happens to the germline. So the way to think about it is we are actually having a cell non-autonomous signaling here where we affect one tissue, but we get a response in a different tissue. And the advantage of this is that if it's blind to sarcopenia or polyglutamine diseases or Alzheimer's or so on and so forth, then we don't care what the disease or the problem the organism is dealing with. We might have a way to tell the organism, do better, or maintain your ability to maintain your somatic tissues. So with that, I will finish. This is Neta, that sadly is not here. And thank you, and I will take questions.
Also, please, questions for Hagai. Sorry, Hagai, I just slipped my mind.
No question. Everybody is. Okay.
So, thank you very much. Thank you, everybody. Perfect timing. Quarter an hour, we have our exciting meeting.